Can human beings be cloned?
In theory, yes. There are, however, many technical obstacles to human cloning, as well as moral, ethical, philosophical, religious, and economic issues to be resolved before a human being could be cloned. At the present time, most scientists would agree that cloning a human being is unsafe under current conditions.
Do animal societies have a culture?
Culture can be defined as the set of societal rules that are passed from one generation to the next. Parents or caregivers teach juveniles what they need to know in order to participate in their society. This phenomenon has been noted among non-human primates and elephants as well as other species. Elephant families rely on memory and the knowledge of the matriarch. Oldest female, to respond to social cues. The matriarch controls the direction in which the herd moves, and where and for how long feeding occurs. When there is danger, the other herd members cluster around the matriarch. Perhaps most striking is the tender loving care lavished on young elephants. The bond between mother and daughter elephants lasts up to 50 years.
What is ELSI as it pertains to the Human Genome Project?
ELSI is a specific aspect of the Human Genome Project that deals with the ethical, legal, and social, issues, arising from increasing availability of genetic information. About 3 to 5% of the budget of the HGP is devoted to ELSI. It represents the largest bioethics study in the world.
What are Turgor movements?
Turgor movements in plants are movements that are reversible. These movements are caused by changes in the turgor pressure of specific cells. For example, some plants exhibit different leaf positions during the day than at night.
What is the relationship between probability and genetics?
Probability is a branch of mathematics used to predict the likelihood of an event occurring. It is also an important tool for understanding inheritance patterns of specific traits. There are two rules that are applied to genetic inheritance. 1. The rule of 388 multiplication is used when determining the probability of any two events happening simultaneously. For example, if the probability of having dimples is 1/4, and the probability of having a male child is 1/2, then the probability of having a boy with dimples is 1/4 x 1/2, or 1/8. 2. The rule of addition is used when determining the outcome of an event that can occur in two or more ways. For example, if we were to consider the probability of having a male child or of having a child with dimples, then the probability would be 1/2 plus 1/4, or 3/4. In other words, chances are 3 out of 4 that a given birth will produce a male child or a child with dimples.
How many antimicrobial products are available?
More than 8,000 antimicrobial products are registered with the U.S. Environmental Protection Agency, EPA. The EPA regulates products that kill microbes on inanimate surfaces. The various products contain more than 300 different active ingredients and are available as sprays, liquids, concentrated powders, and gases. More than 50% of the 8,000 antimicrobial products are used to control infectious microorganisms in healthcare settings.
In the life cycles of seedless, vascular plants such as ferns, is the sporophyte or gametophyte dominant?
The seedless, vascular plants exhibit an alternation of generations consisting of heteromorphic diploid and haploid phases, as found in all plants. The life cycle of seedless, vascular plants is dominated by the sporophyte. The sporophyte is the plant, for example, trees and flowers that everyone visualizes when they think of plants.
How dangerous is Clostridium botulinum?
The bacterium Clostridium botulinum can grow in food products and produce a toxin called botulinum, the most toxic substance known. Microbiologists estimate that 1 gram of this toxin can kill 14 million adults. This bacterium can withstand boiling water, 212 degrees Fahrenheit or 100 degrees Celsius, but is killed in 5 minutes at 248 degrees Fahrenheit 120 degrees Celsius. This tolerance makes Clostridium botulinum a serious concern for people who can vegetables at home. If home canning is not done properly, this bacterium will grow in the anaerobic conditions of the sealed container and create extremely poisonous food. The endospores of Clostridium botulinum can germinate in poorly prepared canned goods. So individuals should never eat food from a can that appears swollen, as it is a sign that the can has become filled with gas released during germination. Consuming food from a can containing endospores that have undergone germination can lead to nerve paralysis, severe vomiting, and even death.
What is a gene gun?
The gene gun, developed by Cornell University plant scientists, is a method of direct gene transfer that is used in plant biotechnology. In order to transfer genes into plants, gold or tungsten microspheres, 1 micrometer in diameter, are coated with DNA from a specific gene. The microspheres are then accelerated toward target cells, contained in a petri dish, at high speed. Once inside the target cells, the DNA on the outside of the microsphere is released and can be incorporated into the plant's genome. This method is also known as microprojectile bombardment or biolistics. The survival rate of bombarded cells varies with the rate of penetration. For example, if the particle penetration rate reaches 21 per cell, approximately 80% of bombarded cells may die.
Examples of products and organisms made in bioreactors. What is a chimera?
The chimera from Greek mythology is a fire-breathing monster with a lion's head, goat's body, and a serpent's tail. The chimera of biotechnology is an animal formed from 2 different species or strains, that is, a mixture of cells from 2 very early embryos. Most chimeras used in research are made from different mouse strains. Chimeras cannot reproduce.
What is meant by the terms homozygous and heterozygous?
Since each diploid organism has 2 alleles for every gene, the alleles can be alike, homozygous, or different, heterozygous. These terms are derived from the Greek terms homos, meaning same, and heteros, meaning different, plus the Greek suffix zygotos, meaning yoked together.
What is the function of the endocrine system?
The endocrine system is the main chemical regulating system of an organism. Hormones, chemicals made and secreted by endocrine glands or neurosecretory cells, are the main messengers of the endocrine system. Hormones are transported in the blood to all parts of the body and interact with target cells. Cells that contain hormone receptors, and they regulate metabolic rate, growth, maturation, and reproduction.
What is polyploidy?
Polyploidy is a condition where complete, extra sets of chromosomes are contained within cells. Polyploid individuals are described as being 3N, triploid, 4N, tetraploid, and so on. Humans cannot survive as polyploids and rarely as aneuploids.
What are the 3 common types of leaf arrangement on a stem?
The 3 most common types of leaf arrangement are, 1. Alternate, or spiral, 2. Opposite, and 3. Whorled. In many plants the leaves are arranged in a pattern alternating on either side of the stem. In other plants the leaves are opposite each other in pairs on the stem. In still other plants the leaves are whorled with 3 or more attached to one level of the stem.
What is the relationship between lichens and air pollution?
Lichens are extremely sensitive to pollutants in the atmosphere and can be used as bioindicators of air quality. They absorb minerals from the air, from rainwater, and directly from their substrate. Lichen growth has been used as an indicator of air pollution, especially sulfur dioxide. Pollutants are absorbed by lichens, causing the destruction of their chlorophyll, which leads to a decrease in the occurrence of photosynthesis and changes in membrane permeability. Pollutants upset the physiological balance between the fungus and the alga or cyanobacterium, and degradation or destruction of the lichens results. Lichens are generally absent in and around cities, even though suitable substrates exist. The reason for this is the polluted exhaust from automobiles and industrial activity. They are beginning to disappear from national parks and other relatively remote areas that are becoming increasingly contaminated by industrial pollution. The return of lichens to an area frequently indicates a reduction in air pollution.
How does ergot affect humans and cattle?
Eating bread and other grain products contaminated with ergot causes the disease called St. Anthony's fire. Common during the Middle Ages, this disease, which causes sensations of intense heat followed by a complete loss of sensation in an infected person's limbs, no longer occurs very frequently due to improved techniques of grain production and milling. Cattle that graze on grains infected with ergot are able to ingest enough to cause death or the spontaneous abortion of fetuses.
What features of reptiles enabled them to become true land vertebrates?
Legs were arranged to support the body's weight more effectively than in amphibians, allowing reptile bodies to be larger and to run. Reptilian lungs were more developed with a greatly increased surface area for gas exchange than the sac-like lungs of amphibians. The three-chambered heart of reptiles was more efficient than the amphibian heart. In addition, the skin was covered with hard, dry scales to minimize water loss. However, the most important evolutionary adaptation was the amniotic egg, in which an embryo could survive and develop on land. The eggs were surrounded by a protective shell that prevented the developing embryo from drying out.
What are some examples of genetic engineering in animals and microbes?
One of the earliest applications of biotechnology was the genetic engineering of a growth hormone. Bovine GH, produced naturally in the bovine pituitary. Bovine GH can increase milk production in lactating cows. Using biotechnology, scientists bioengineered the gene that controls bovine GH production into E. coli, grew the bacteria in fermentation chambers, and thus produced large quantities of bovine GH. The bioengineered bovine GH, when injected into lactating cows, resulted in an increase of up to 20% in national milk production. Using bovine GH, farmers are able to stabilize milk production in their herds, avoiding fluctuations in production levels. A similar regimen was adapted using the pig equivalent of growth hormone, porcine GH. Injected in pigs, porcine GH reduced back fat and increased muscle, meat, gain. The first transgenic animal available as a food source on a large scale was the salmon, which reached U.S. food markets in 2001, following rigid evaluations of consumer and environmental safety. These salmon have the capability of growing from egg to market size, 6 to 10 pounds, in 18 months. As compared to conventional fish breeding, which takes up to 36 months to bring a fish to market size. The use of transgenic salmon can help reduce overfishing of wild salmon stocks.
What is a variable?
A variable is something that is changed or altered in an experiment. For example, to determine the effect of light on plant growth. Growing one plant in a sunny window and one in a dark closet will provide evidence as to the effect of light on plant growth.
Is obesity inherited?
Genetic clues to obesity have been studied in mice, where at least two genes, obese, OB, and diabetes, DB, have been identified and analyzed. The OB gene codes for the weight-controlling hormone leptin. From the Greek term leptos, meaning slender, which is produced by fat cells. Once leptin is released by fat cells, it travels to the hypothalamus, a specific area of the brain, where it binds to receptors. The DB gene codes for these hypothalamic receptors. Once leptin is bound, it regulates the rate of energy consumption. However, body weight is a complex phenotype, and it is likely that there are more genes to be found that control body weight.
How do rodents, rabbits, and hares digest cellulose?
Unlike cows, which have a rumen to digest cellulose, rodents, rabbits, and hares have a cecum, a large pouch to digest cellulose with the assistance of microorganisms. The cecum is located at the junction between the small and large intestines. It is impossible for these animals to regurgitate the contents of their stomachs. Like ruminants, because the cecum is located beyond the stomach. Instead, these animals pass their food through their digestive tract a second time by ingesting their feces. When feces pass through the digestive tract, it is possible for these animals to absorb the nutrients produced by the microorganisms in the cecum.
How does kidney dialysis remove waste products from the body?
Damaged or diseased kidneys are not capable of removing toxic waste substances from the body. Kidney dialysis removes the nitrogenous waste and regulates the pH of the blood when the kidneys do not work. Blood is pumped from an artery through a series of tubes made of a permeable membrane and a dialysing solution. Urea and excess salts diffuse out of the blood as it circulates through the dialysing machine and are then discarded. Necessary ions diffuse from the dialysing solution back into the blood. The cleansed blood is then returned to the body.
What are biosensors?
A biosensor is a unique combination of biological substances, e.g., microbe, cell, enzyme, antibody, linked to a detector. It can be used to measure very low concentrations of a particular substance. An example of a biosensor currently on the market is the insulin pump, which maintains correct blood glucose concentrations for diabetics.
What is the life cycle of a typical fungus such as the black bread mold, Rhizopus nigricans?
The life cycle of black bread mold, Rhizopus nigricans, is characteristic of the members of the phylum Zygomycota. This fungus has a period of sexual reproduction and a period of asexual reproduction that occurs more frequently. During sexual reproduction, there is a fusion of gametangia. The resulting zygosporangium forms a thick coat that awaits favorable conditions in order to conduct further development. When conditions are favorable, the zygosporangium germinates into a sporangium. During asexual reproduction, spores are produced in the sporangium and then dispersed.
Do fungi only decompose dead and decaying organic matter?
Not only do fungi consume dead and decaying organic matter, but some attack living plants and animals as they serve as a source for necessary organic molecules. Fungi often cause diseases among plants and animals. They are some of the most harmful pests to living plants and are responsible for billions of dollars in agricultural losses each year. Food products that have been harvested and stored are not immune to fungal decay. Fungi often secrete substances into the foods they attack, making the foods unpalatable or even poisonous.
What kinds of molecules are hormones?
There are two basic types of hormones, those that are water-soluble, hydrophilic, and those that are not, hydrophobic. Hydrophilic hormones include those derived from amino acids, peptides, and proteins. The hydrophobic group includes steroids, which are derived from cholesterol. Steroid hormones include testosterone, estrogen, cortisol, and aldosterone.
What is meiosis?
Meiosis is often referred to as reduction division, meaning that the number of chromosomes present is reduced from 2n, diploid, to n, haploid. The meiotic process consists of two separate cell divisions and occurs in the gonads, ovaries and testis. It is most important to sexual reproduction because of genetic variation that occurs as a result of this process.
What is the definition of behavior?
In its broadest sense, behavior covers all kinds of movement and responses to environmental changes. In other words, behavior is the term used to describe what an animal does.
Which birds lay the largest and smallest eggs?
The elephant bird, Epiornis maximus, an extinct flightless bird of Madagascar. Also known as the giant bird or roc, laid the largest known bird eggs. Some of these eggs measured as much as 13.5 in, 34 centimeters, in length and 9.5 in, 24 centimeters, in diameter. The largest egg produced by any living bird is that of the North African ostrich, Struthiocamelus. The average size is 6 to 8 in, 15 to 20.5 centimeters, in length and 4 to 6 in, 5 to 15 centimeters, in diameter. The smallest mature egg, measuring less than 0.39 in, 1 centimeter, in length, is that of the vervain hummingbird, Melissuga minima, of Jamaica. Generally speaking, the larger the bird, the larger the egg. However, when compared with the bird's body size, the ostrich egg is one of the smallest eggs, while the hummingbird's egg is one of the largest. The kiwi bird of New Zealand lays the largest egg relative to body size of any living bird. Its egg weighs up to 1 pound (0.5 kilograms).
Where is RNA formed?
All RNA is formed in the nucleus, eukaryotic cells, or in the nucleoid region, prokaryotic cells. The principal enzyme responsible for RNA synthesis is RNA polymerase.
What is the fastest growing land plant?
Bamboo, Bambusa spp. Native to tropical and subtropical regions of Southeast Asia and islands of the Pacific and Indian Oceans. Is the plant that gains height most quickly. Bamboo can grow almost 3 feet 1 meter in 24 hours. This rapid growth is produced partly by cell division and partly by cell enlargement.
How many different animals are there?
Biologists have described and named more than 1 million species of animals. And some biologists believe that there are several million to 10 million more species that remain to be discovered, classified, and named.
How do animals recognize each other?
We know that animals can use scent, color, and sound to recognize individuals. And they may also be able to recognize other attributes as well. A recent study on sheep intelligence indicates that easily herded animals may be smarter than originally thought. The sheep were shown pictures of other sheep and were subsequently rewarded if they moved toward a selected picture. The sheep learned which face produced a reward. Ultimately, it was shown that sheep were able to pick a selected picture 80% of the time and could remember up to 50 images for 2 years.
What was Camillo Golgi's, 1843 to 1926, contribution to histology?
In 1873 Camillo Golgi, 1843 to 1926, devised a way to stain tissue samples with inorganic dye using silver salts. When he applied this technique and stained nerve tissue, he was able to see details previously not visible.
How does a cell respond to injury?
In most tissues, injured cells die and are subsequently replaced. However, in the case of nervous tissue, where dead cells cannot be replaced, a number of events can occur. Nerve growth factor is produced by adjoining neurons. This can induce sprouting and growth of previously dormant neurons.
What roles do fungi play in recycling?
Fungi play a key role in the recycling of many elements. As the primary decomposers in the biosphere, they break down organic matter, including dead plants and other vegetation. As fungi actively decompose materials, carbon, nitrogen, and the mineral components present in organic compounds are released, these elements can all be recycled. During decomposition, carbon dioxide is released into the atmosphere and minerals are returned to the soil. It is estimated that, on average, the top 8 in 20 centimeters of fertile soil contain nearly 5 metric tons of fungi and bacteria per 2.47 acres, 1 hectare. Without fungi acting as decomposers, dead organic matter would overpower the world and life on Earth would eventually become impossible.
Are there more marine or freshwater sponges?
There are approximately 5,000 species of marine, saltwater, sponges and 150 species of freshwater sponges.
Which scientific disciplines provide evidence for evolution?
Although information from any area of natural science is relevant to the study of evolution, there are several in particular that directly support the work of Darwin and Wallace. Paleobiology, geology, and organic chemistry provide insight on how living organisms have evolved. Ecology, genetics, and molecular biology also demonstrate how living species are currently changing in response to their environments and therefore undergoing evolution.
How many different organisms have been identified by biologists?
Approximately 1.5 million different species of microorganisms, plants, and animals have been described and formally named. Some biologists believe this is only a fraction of species that exist, estimating that there are more than 10 million species waiting to be discovered, classified, and named. It is estimated that 15% of all species are marine organisms. Most scientists agree that only 5% of bacteria, fungi, nematode, and mite species have been discovered.
How do we know that DNA is antiparallel?
The central structure of a DNA molecule is the deoxyribose molecule, which has 5 carbons, each of which is numbered. At one end, phosphate can only be attached at the number 5 carbon, 5. At the other end, there is no phosphate attached, and this is called the number 3 carbon. 3, and, here, a hydroxyl group is attached to the sugar instead of a phosphate.
Why is there such a range of skin color in humans?
Studies on the genetic components of skin color indicate that at least 3 and perhaps as many as 4 gene pairs interact to produce a given shade of skin color. 4 gene pairs would generate 4x4x4x4, 256, possible genotypes, which is ample for the number of color variations found among humans.
What is the cellular fountain of youth?
Research on telomeres and telomerase shows that cells can be kept alive far beyond their normal lifespan. Specific studies have been carried out on the roundworm, C. elegans, in which longer lifespans were observed in worms with longer telomeres.
What are some examples of fermented foods?
Fermented foods are foods produced by microorganisms. A combination of bacteria and fungi are used in the fermentation process of many food products. Examples of fermented foods are cheese, sour cream, yogurt, sauerkraut, vinegar, olives, pickles, soy sauce, miso, chocolate, coffee, and most alcoholic beverages.
What is the Darwin-Wallace theory?
The Darwin-Wallace theory can be summarized as the following. Species as a whole demonstrate descent with modification from common ancestors. And natural selection is the sum of the environmental forces that drive those modifications. The modifications or adaptations make the individuals in the population better suited to survival in their environment, more fit as it were. The 4 postulates presented by Darwin in On the Origin of Species by Natural Selection are as follows. 1. Individuals within species are variable. 2. Some of these variations are passed on to offspring. 3. In every generation more offspring are produced than can survive. 4. The survival and reproduction of individuals are not random, the individuals who survive and go on to reproduce the most are those with the most favorable variation. They are naturally selected. It follows logically from these that the characteristics of the population will change with each subsequent generation, until the population becomes distinctly different from the original. This process is known as evolution.
How did the original Celsius temperature scale differ from the one in use now?
In 1742 the Swedish astronomer Anders Celsius, 1701–1744, set the freezing point of water at 100 degrees Celsius and the boiling point of water at 0 degrees Celsius. It was Carolus Linnaeus, 1707 to 1778, who reversed the scale. But a later textbook attributed the modified scale to Celsius, and the name has remained.
What is gene redundancy?
Gene redundancy refers to having multiple copies of the same gene. This assures that essential genes that are required in large amounts, like those for ribosomal RNA, rRNA, can be transcribed at multiple sites. In addition, gene redundancy provides for a source of gene sequences that can be modified by natural selection without immediately destroying the organism.
What is the average size of a virus?
Viruses are much smaller than bacteria. The smallest viruses are about 17 nanometers in diameter, and the largest viruses are up to 1000 nanometers, 1 m, in length. By comparison, the bacterium Escherichia coli is 2000 nanometers in length. A cell nucleus is 2800 nanometers in diameter, and an average eukaryotic cell is 10,000 nanometers in length.
Which protist is an indicator of polluted water?
Euglenoids are unicellular flagellates. Many euglenoids are capable of photosynthesis and are autotrophic. They are commonly found in freshwater ponds and puddles. Others do not carry on photosynthesis and are heterotrophic. Often found in water with large amounts of organic material. Euglenoids frequently serve as bioindicators and are found in large numbers in polluted waters.
What is the ecological role of lichens?
Lichens account for approximately 8% of the vegetation covering Earth's surface. In certain environments, such as regions of tundra, they cover vast areas of land. Lichens delay global warming by consuming significant amounts of carbon dioxide. CO2, during photosynthesis. When they cover the ground, they prevent soil from drying out. In desert areas they are able to capture and conserve the moisture present in fog and dew. Lichens release nutrients, such as nitrogen and phosphorus, which are important in regions with nutrient-poor soils as the nutrients aid tree growth. Lichens are also an important food source for many species of animals, including wild turkeys and reindeer of the Arctic tundra. Birds such as the olive-headed weaver of Madagascar and the goldfinch of Europe use lichens to build their nests.
Can cells ever change jobs?
the more specialized of a function a cell performs, the less likely it is for the cell to change jobs within an organism. However, there are some cells that have unspecialized functions and are able to adapt to the changing needs of the body. In mammals, a good example of cells with adapting functions would be bone marrow cells, which are responsible for producing different types of cells in the blood. Bone marrow cells produce red blood cells and 5 types of white blood cells. Slime molds of the kingdom Protista have cells that are capable of drastically changing their function. The cellular adaptations that can occur in slime molds allow them to change from single-celled amoebas to multicellular, reproductive spore producers.
How do T cells differ from B lymphocytes?
Lymphocytes are one variety of white blood cells and are part of the body's immune system. The immune system fights invading organisms that have penetrated the body's general defenses. T cells, responsible for dealing with most viruses, for handling some bacteria and fungi, and for cancer surveillance, are one of the 2 main classes of lymphocytes. T lymphocytes, or T cells, compose about 60 to 80% of the lymphocytes circulating in the blood. They have been educated in the thymus to perform particular functions. Killer T cells are sensitized to multiply when they come into contact with antigens, foreign proteins. On abnormal body cells, cells that have been invaded by viruses, cells in transplanted tissue, or tumor cells. These killer T cells attach themselves to the abnormal cells and release chemicals, lymphokines, to destroy them. Helper T cells assist killer cells in their activities and control other aspects of the immune response. When B lymphocytes, which compose approximately 10 to 15% of total lymphocytes, contact the antigens on abnormal cells, the lymphocytes enlarge and divide to become plasma cells. Then the plasma cells secrete vast numbers of immunoglobulins or antibodies into the blood, which attach themselves to the surfaces of the abnormal cells, to begin a process that will lead to the destruction of the invaders.
Did dinosaurs and humans ever coexist?
No. Dinosaurs first appeared in the Triassic period, about 220 million years ago, and disappeared at the end of the Cretaceous period, about 65 million years ago. Modern humans, Homo sapiens, appeared only about 25,000 years ago. Movies that show humans and dinosaurs existing together are only Hollywood fantasies.
What are the specialized cells in plants?
All plant cells have several common features, such as chloroplasts, a cell wall, and a large vacuole. In addition, a number of specialized cells are found only in vascular plants. They include parenchyma cells, parenchyma, from the Greek para, meaning beside, and enchine, meaning to pour in. Cells are the most common cells found in leaves, stems, and roots. They are often spherical in shape with only primary cell walls. Parenchyma cells play a role in food storage, photosynthesis, and aerobic respiration. They are living cells at maturity. Most nutrients in plants, such as corn and potatoes, are contained in starch-laden parenchyma cells. These cells comprise the photosynthetic tissue of a leaf, the flesh of fruit, and the storage tissue of roots and seeds. Collenchyma cells, collenchyma, from the Greek term kolla, meaning glue. Cells have thickened primary cell walls and lack secondary cell walls. They form strands or continuous cylinders just below the surfaces of stems or leaf stalks. The most common function of collenchyma cells is to provide support for parts of the plant that are still growing, such as the stem. Similar to parenchyma cells, collenchyma cells are living cells at maturity. Sclerenchyma cells, sclerenchyma, from the Greek term skleros, meaning hard. Cells have tough, rigid, thick secondary cell walls. These secondary cell walls are hardened with lignin, which is the main chemical component of wood. It makes the cell walls more rigid. Sclerenchyma cells provide rigid support for the plant. There are two types of sclerenchyma cells, fiber and sclereid. Fiber cells are long, slender cells that usually form strands or bundles. Sclereid cells, sometimes called stone cells, occur singly or in groups and have various forms. They have a thick, very hard secondary cell wall. Most sclerenchyma cells are dead cells at maturity. Xylem, xylem, from the Greek term xylos, meaning wood, is the main water-conducting tissue of plants and consists of dead, hollow, tubular cells arranged end to end. The water transported in xylem replaces that lost via evaporation through stomata. The two types of water-conducting cells are tracheids and vessel elements. Water flows from the roots of a plant up through the shoot via pits in the secondary walls of the tracheids. Vessel elements have perforations in their end walls to allow the water to flow between cells. Phloem, the two kinds of cells in the food-conducting tissue of plants, the phloem, from the Greek term phloios, meaning bark, are sieve cells and sieve tube members. Sieve cells are found in seedless vascular plants and gymnosperms, while sieve tube members are found in angiosperms. Both types of cells are elongated, slender, tube-like cells arranged end to end with clusters of pores at each cell junction. Sugars, especially sucrose, other compounds, and some mineral ions move between adjacent food-conducting cells. Sieve tube members have thin primary cell walls but lack secondary cell walls. They are living cells at maturity. Epidermis. Several types of specialized cells occur in the epidermis, including guard cells, trichomes, and root hairs. Flattened epidermal cells, one layer thick and coated by a thick layer of cuticle. Cover all parts of the primary plant body.
What setbacks have occurred in the use of gene therapy?
During the 1990s there were more than 4,000 patients involved in gene therapy trials. Unfortunately, most of these trials were not successful. And a further setback occurred in 1999 with the death of 18-year-old Jesse Gelsinger. Gelsinger suffered from ornithine transcarbamylase deficiency, a rare metabolic disease in which toxic levels of ammonia can be produced by the liver. He died of complications due to a severe immune response to the viral carrier used to deliver the therapeutic genes.
Which cell structures are unique to plant cells?
The chloroplast, central vacuole, tonoplast, cell wall, and plasmodesmata are only found to occur in plant cells.
Which part of the wheat plant, Triticum aestivum, is used to make flour?
Wheat is a monocotyledon grass whose fruit, the grain, or kernel, contains one seed. The endosperm and embryo of the wheat plant are surrounded by the pericarp, or fruit wall, and the remains of the seed coat. More than 80% of the volume of the wheat kernel is made up of the starchy endosperm. White flour is made by milling the starchy endosperm. Wheat bran constitutes approximately 14% of the wheat kernel and is found in the covering layers and the outermost layer of the kernel, called the alurone layer. Wheat germ is the embryo of the wheat plant and represents approximately 3% of the wheat kernel. There are nearly 24 species of wheat. The most important ones for commercial use are common wheat, Triticum aestivum, sometimes referred to as Triticum vulgar, and durum wheat, Triticum durum. The varieties of common wheat account for 90% of the wheat grown worldwide. Durum wheat accounts for 5 to 7% of the wheat grown, and all other species account for the remainder of the wheat grown.
What was the first professional organization of physiologists?
The first organization of physiologists was the Physiological Society, founded in 1876 in England. In 1878 the Journal of Physiology began publication as the first journal dedicated to reporting results of research in physiology. The American counterpart, the American Physiological Society, was founded in 1887. The American Physiological Society's sponsored publication, the American Journal of Physiology, began in 1898.
How does the nervous system of invertebrates differ from that of vertebrates?
The least complex nervous system is the nerve net of cnidarians such as hydras. The nerve net is a network of neurons located throughout the radially symmetric body. The neurons are in contact with one another and with muscle fibers within epidermal cells. These animals lack a head and brain. Invertebrates that display bilateral symmetry such as planarians, annelids, and arthropods have a brain, a concentration of neurons at the anterior or head end, and one or more nerve cords and the presence of a central nervous system. Vertebrates have a central nervous system and a peripheral nervous system.
How much of the human genome actually codes for genetic information?
Only about 5% of the human genome appears to code for genetic information.
What are the macronutrients and micronutrients of plants?
The macronutrients of plants are carbon, hydrogen, oxygen, nitrogen, potassium, calcium, phosphorus, magnesium, and sulfur. These all are nearly or in some cases far greater than 1% of the dry weight of a plant. The micronutrients are iron, chlorine, copper, manganese, zinc, molybdenum, and boron. Each of the micronutrients constitutes less than 1 to several hundred parts per million in plants. Sodium, silicon, cobalt, and selenium are beneficial elements. Research has not shown that these elements are essential for plant growth and development.
How are nitrogenous wastes metabolized?
Ammonia, urea, and uric acid are nitrogenous waste products. They are the result of the breakdown of various molecules including nucleic acids and amino acids. Some amino acids are used for new protein synthesis, while others are oxidized to generate energy or converted to fats or carbohydrates that can be stored. Once broken down, the amino group NH2, containing 1 nitrogen and 2 hydrogens, must be removed or the animal will eventually be poisoned. Ammonia is the most toxic of the nitrogenous wastes. It is formed by the addition of a third hydrogen atom to the NH2 group. Both urea and uric acid are less toxic forms of nitrogenous waste but have greater energy requirements for their production.
What is Bayesian mimicry?
In 1861 Henry Walter Bates, 1825 to 1892, a British naturalist, proposed that a non-toxic species can evolve, especially in color and color pattern, to look or act like a toxic or unpalatable species in order to avoid being eaten by a predator. The classic example is the viceroy butterfly, which resembles the unpalatable monarch butterfly. This is called Batesian mimicry. Bates is also well known as a colleague of Alfred Russel Wallace. In fact, it was Bates who introduced Wallace to botany and field collecting of animals and plants.
What is germline therapy?
Germline therapy involves gene alteration within the gametes, eggs or sperm, of an organism. Alterations in gamete genes transfer the gene to all cells in the next generation. As a result, all members of future generations will be affected.
What methods of analysis are used for DNA profiling?
Both RFLP, restriction fragment length polymorphism, and PCR, polymerase chain reaction, techniques can be used for DNA profiling. The success of either method depends on identifying where the DNA of two individuals varies. The most and how this variation can be used to discriminate between two different DNA samples. Since RFLP uses markers in regions that are highly variable, it is unlikely that the DNA of two unrelated individuals will be identical. However, this method requires at least 20 nanograms of purified, intact DNA. PCR-based DNA fingerprinting is a rapid, less expensive method and requires only a very small amount of DNA. As little as 50 white blood cells.
Are humans still evolving?
In order for humans to continue evolving in a measurable way, we would predict that those individuals with certain variations, alleles, of traits must reproduce more efficiently than those without those alleles, such that eventually the alleles in question become more common in the population. For example, if nearsighted individuals were less likely to produce healthy offspring, for example, perhaps they have trouble finding enough food, then we would expect that eventually there would be very few nearsighted individuals in the population, assuming that nearsightedness is genetic. Because of our uniquely human skill of inventing things like eyeglasses and corrective surgery, which counteract genetic programming without actually changing the genes, there is little selective pressure against being nearsighted, so it is unlikely to be lost from the human population gene pool. The same could be said for many other traits. So while our gene pool could certainly change with time, it is unclear whether it will change in a clearly measurable direction such that we physically evolve.
What is the difference between poison ivy, oak, and sumac?
These North American woody plants grow in almost any habitat and are quite similar in appearance. Each variety of plant has 3 leaf compounds that alternate, berry-like fruits, and rusty brown stems. Poison ivy, Rus radicans, grows like a vine rather than a shrub and can grow very high, covering tall, stationary items such as trees. The fruit of our radicans is gray in color and is without hair, and the leaves of the plant are slightly lobed. Rus toxicodendron, commonly known as poison oak, usually grows as a shrub, but it can also climb. Its leaflets are lobed and resemble the leaves of oak trees, and its fruit is hairy. Poison sumac, Rus vernix, grows only in acidic, wet swamps of North America. This shrub can grow as high as 12 feet 3.6 meters. The fruit it produces hangs in a cluster and ranges from gray to brown in color. Poison sumac has dark green leaves that are sharply pointed, compound, and alternating. It also has inconspicuous flowers that are yellowish green. All parts of poison ivy, poison oak, and poison sumac can cause serious dermatitis.
What are archaebacteria?
Archaebacteria, domain Archaea, are primitive bacteria that often live in extreme environments. This domain includes the following: 1. Thermophils, heat lovers, which live in very hot environments, including the hot sulfur springs of Yellowstone National Park, which reach temperatures ranging from 140 to 176 degrees Fahrenheit, 60 to 80 degrees Celsius. 2. Halophils, salt lovers, which live in locations with high concentrations of salinity, such as the Great Salt Lake in Utah, which has salinity levels that range from 15 to 20%. Seawater normally has a level of salinity of 3%. 3. Methanogens, which obtain their energy by using hydrogen gas, H2, to reduce carbon dioxide, CO2, to methane gas, CH4.
What is the difference between a fat and a lipid?
Lipids are bioorganic molecules that are hydrophobic. In other words, they do not mix with or dissolve in water. Among lipids there is a category known as fats. Each fat molecule is comprised of a glycerol, alcohol molecule and at least one fatty acid, hydrocarbon chain with an acid group attached.
What is unusual about cave moss?
Cave moss, Schistostigma pennata, is a small plant with reflective, subspherical cells at 166 its tips. These cells give off an eerie glow that is gold and green in color. In Japan, the plant has been the subject of numerous books, television shows, newspaper and magazine articles, and even an opera. There is a national monument to this species near the coast of Hokkaido, where it grows near a small cave.
Do bacteria all have the same shape?
Bacteria have three main shapes: spherical, rod-shaped, and spiral. Spherical bacteria, known as cocci, occur singularly in some species and as groups in other species. Cocci have the ability to stick together and form a pair, diplococci, when they stick together in long chains. They are called streptococci. Irregularly shaped clumps or clusters of bacteria are called staphylococci. Rod-shaped bacteria, called bacilli, occur as single rods or as long chains of rods. Spiral or helical-shaped bacteria are called spirilla.
What is the relationship between blue jays, monarch butterflies, and milkweed?
As part of their life cycle, female monarchs lay their eggs exclusively on milkweed plants. After a few days, the eggs hatch and a yellow, black, and white striped caterpillar emerges from each egg. These caterpillars are totally dependent on milkweed plants. Although the plants contain toxic substances, cardinalides, that are poisonous to other animals, the toxin is harmless to the monarch. Blue jays, who spend much of their day searching for food, will often eat insects such as adult monarch butterflies to supplement their otherwise vegetarian diet. However, if the food tastes bad, the blue jays will vomit up the food and will then learn individually to avoid the food in the future. Thus, wild monarch butterflies with high levels of cardinalide concentrations are less susceptible to natural predation by birds. This is an example of operant conditioning in the wild.